So the topic for this week is protein methods, which is kind of a huge topic. It's very broad. There's a lot of protein methods. So what I'm going to do is kind of do just like very broad overview of different techniques you can use with proteins for different purposes. It's like kind of get the impression a lot of you have done some DNA stuff already. It seemed like you've done a lot, well, some PCR and things like that. But has anybody worked with proteins very much? Okay. Maybe done a Western blot or anything like that? Sort of. Okay. Well, they're good. They're good to know about if you use proteins in your research in the future. So to get started, there's this slide, which referred to as the central dogma of molecular biology. DNA is transcribed into RNA, which is translated into protein. So you can't have proteins without DNA. If you have DNA without proteins, there's no point to the DNA. So they're equally important. Some people tend to put themselves in one camp or the other. They work only with DNA or they work only with proteins. So as a lot of you are familiar with sort of the DNA, RNA techniques, there are lots of them for different purposes. We have things like gel electrophoresis, which conveniently Joanna talked about last week, and PCR. RNA sequencing, digests, DNA sequencing, cloning, genomics, transfection, like wide range of different techniques used for very different purposes, but all based around DNA nucleic acids. So southern or northern blots. Proteins have a similarly wide array of techniques you can use. Listed some of them out here. I'm going to mention probably most of these, at least by name, but I won't go in crazy detail about what they all are for. Some of them are meant for protein detection, some are meant for quantification, some are meant for analyzing proteins, like the structures of proteins and the sequences of them. So this is kind of where we're starting at, is a huge variety of techniques to touch on, at least briefly. So protein review. Has anybody had to memorize the amino acids yet? Nobody? Really? For nothing? Oh, man. Well, so there's 20 of them. I feel like I had to do it for like 4 different classes. It's crazy. But so these are the building blocks of proteins. Linked together, they make polypeptides, which fold into the shape of a protein. All of the amino acids have different side chains. They have the same basic structure, but each one has different functional groups coming off of it. This slide has kind of grouped them into aliphatic, aromatic, acidic, basic. Like they can be grouped into different categories, and they behave differently. Some of them are charged, some of them are polar, some of them are nonpolar, and that can affect both how the protein folds. So for example, hydrophobic amino acids want to be in contact with more hydrophobic acids, and so that can affect the fold. They're also really important for actually functions of proteins, so functions of enzymes. Sometimes there's a key amino acid in the active site of the enzyme that makes it work the way it does. So the amino acid sequence of a protein is super important, and each protein has a sort of distinct amino acid sequence. So that's what this is showing. You link them together by what are called these peptide bonds. You have a peptide backbone, which is sort of a chain of C's, of carbons and nitrogens, and then the side chains will hang off. So yeah, there's a couple of them on there. But yeah, peptide bonds linking amino acids together, and that is a polypeptide chain. So we can describe sort of 4, this might also be your view, I don't know, but 4 kind of categories of protein structure starting at the base level of amino acid sequence. So the amino acid sequence, the order in which the amino acids are laid out in a protein, is called the primary structure of the protein. The secondary structure is the structure that forms when amino acids form basically hydrogen bonds with each other. So they form these kind of not macromolecular structures, but sort of structures within themselves. They form basically 2 different types called pleated sheets, also called beta sheets, or alpha helices, which are sort of spiral-like domains. That's called the secondary structure. Tertiary structure is how the whole protein itself folds. So the shape it forms once the whole polypeptide has folded the way it's supposed to kind of looks like a blob, but usually it's very important what that blob actually looks like. So that's the tertiary structure. Quaternary structure is what happens when you get one or more polypeptide chains associated with each other. Usually they end up covalently linked. And so multiple chains linked together is known as the quaternary structure of the protein. So like I said, there's many different purposes for analyzing proteins and many different purposes for the different assays that you use. You can sort of separate them into categories. First one I came up with here, purifying and separating proteins. So if you're trying to analyze a protein, if you're trying to analyze a particular protein or a particular category of proteins, first you want to get it by itself. Usually when you're working with proteins, it's in a mixture of a whole bunch of different ones. Either you're working with whole cells or you're working with tissues. Blood is one thing. Blood contains a whole lot of different proteins, huge variety. So if you want to get a particular protein by itself, there are techniques you can use. But first you have to get them out of wherever they are. So you can't just purify a protein while it's still within a cell, because it doesn't really work that way. So first, for example, you have to break the cell apart. And those are all my different colored polypeptide chains in there, representing different proteins. Some are inside, some are in the membrane. If we want to analyze the ones inside, we have to break open the cell. So you can do that with things like sonicators, which if anybody's ever used one of those, they're kind of hurt your ears if you do it wrong. Detergents can break up the membrane and get the proteins out and solubilize the proteins so that they are then in solution where you can use them. And if you then spin them down, isolate the proteins from the membrane fragments and anything else that might be in the cell that you don't want, you can get your proteins by themselves that way. So example methods, like I said, lysis, meaning breaking cells open if you're using cells. Homogenation, if you're using tissue, so you can put a whole tissue, break that apart and get proteins out that way. Precipitation, you can add certain chemicals to the solution to make the proteins actually fall out of solution. It's mostly if you're doing huge amounts of tissue or things like that, and you spin them down in a very large centrifuge. But yeah, centrifugation, getting the proteins separated from everything else you don't want. Now if you want a specific protein or a specific category of protein out of that mixture that you've made, there are a bunch of different ways to do that too. So this is kind of a general schematic of what a lot of those procedures look like. You start with a protein mixture, a whole bunch of different things together. You want to get, for example, in this one, we want to get the blue and the yellow ones out. So let's say maybe they are very large proteins, even though they don't look like it, but if they're very big, we want to separate the very big ones from the very small ones. So what you can do, kind of in a general sense, you can put proteins onto some kind of column or some kind of, pass them through some kind of medium and separate out the proteins that you don't want. Different ways to do this. There's different ways to separate proteins from each other. Usually, well, a lot of different ways we categorize as chromatography, so separation methods. You can do this through size exclusion, meaning you only end up with proteins above a certain molecular weight or below a certain molecular weight. There are ways to do this passing them through membranes. Like a big protein can't pass through, so it stays on one side, and the small proteins can pass through, so they go to the other side. That's one way. Ion exchange chromatography is another way. If one of your proteins is particularly charged or has a particular affinity for charge, you can separate them that way. Affinity chromatography makes similar use of particular affinities of a protein for something. One sort of subtype of this is immunoprecipitation, so association with an antibody. But if you have a protein that associates particularly strongly with something, you can usually separate it based on that. The HPLC is high performance liquid chromatography. That's a way of separating proteins in according to usually hydrophilic and hydrophobic properties. So proteins that are more hydrophobic will separate out from proteins that are more hydrophilic, and that requires the use of a fancy instrument with liquid pumps and columns and all kinds of stuff. But first few you can actually do kind of on the bench top with a column, directly passing your proteins through and getting the ones you want to retain on there. So purifying, separating, getting the proteins you want out of a mixture. These are examples of that type of method that you might see. There's many more. These are just examples. Another category of purpose you might use protein methods for is detecting and/or quantifying. So determining whether or not a particular protein is in your sample, and quantifying how much of it is there, or quantifying how much protein in general is in your sample. So I've separated these into total protein, which doesn't look at any particular specific protein, just how much protein is in there in general, or specific ones. So, for example, we have 3 different samples here, which I've done each of them in duplicate. You can see that there's different proteins in each well. Some of them have more, some of them have less, but they're just proteins. So if you want to non-specifically detect your proteins, there are a couple different ways you can do that to determine a protein concentration in the sample. 2 of them, one is called the BCA assay, which will give you a purple color with more protein, regardless of what that protein is. The more purple it turns, the more protein you have in your sample. Other one is called Bradford assay, which is a little bit weird because the blank is brown rather than clear, which usually blanks are blank, but this one's brown, and it turns blue with more or less protein. With more protein in your sample will turn more blue, and some variation of brown to blue. So these each have their own advantages and disadvantages. There are certain things they're incompatible with each one. So for example, the BCA assay, if your sample has any kind of reducing agents in it, or things like EDTA, certain things interfere and you can't use a BCA assay, so you'd have to use the Bradford. Similarly, the Bradford you can't use with anything that has detergents, so you would have to use the BCA for that. If you have both reducing agent and detergent, it gets a little complicated, but usually things don't get that messy. So that's for quantifying total protein. Any protein in your sample will react and you can get a total protein concentration. If you're looking for a specific one and you want to detect and quantify that specific protein, usually, often, what happens is you will use kind of an antibody-based technique. You learned about antibodies a couple weeks ago, so that should be helpful. So you can start, for example, with a mixture of proteins in dish, add antibodies, so you can see these are only detecting the red proteins, and then you can quantify the signal from the antibodies that bind. And that's a very general kind of immunoassay. Different ways you can do this. One of them is an ELISA, one's a Western blot. ELISAs, I think, is what were described in the antibody lecture, but nobody's done a Western blot ever? Or kind of, maybe, sort of a little bit? Okay, well that's good. But that's another method that can use antibodies, and that's actually going to come a little bit later. So, apart from detecting and quantifying proteins, you can also analyze the proteins directly. So determine what structure they have, what their amino acid sequence is, what their 3D structure looks like. And these I was not even going to try to make my own pictures of because they're often complicated. So one way to determine the structure of a protein is through X-ray crystallography. So that shoots X-ray beams through a crystallized version of a protein. As you know, it's actually pretty hard to crystallize proteins, so people who do this, that's kind of the hardest part of their experiment, is making the protein crystal. But depending on the structure of the protein, it'll scatter X-rays in different ways, and they hit a detector, come up on a screen, and people who do this know how to interpret that. But that's a way that a lot of the structures of proteins have been determined. So when you see in textbooks and things like that, the big kind of ball and stick structures of proteins, that's oftentimes how they got it, is through something like this. To determine the amino acid sequence of a protein, a lot of times what we use is mass spectrometry, which you end up getting a signal that looks like that. Often what you do is kind of digest the protein, chop it up into small pieces, run them through a mass spectrometer, and then you get peaks depending on the mass to charge ratio of each peptide. So if the peptide is multiply charged, then you have to divide it by multiple charges. But mass to charge ratio, and people who know how this works can interpret something like that and say this is the structure of, this is the amino acid sequence of my protein. So this is an example, that peptide sequence written out there, you can barely see it, but they're single letter abbreviations for amino acids. This spectrum would look like that. Another way that's even more complicated is protein NMR. So I know we have NMR here. I don't know if anybody's used the NMR, but in the chemistry department. We don't do proteins with it because that's also very complicated. But that's another way of determining protein structure. It's a little bit tricky because if there's any kind of change in conformation of the protein over time, it can be hard to see that. But it's another way of determining kind of overall 3D structure of a protein. So different categories of protein assays: quantifying, separating, analyzing the sequence. All of these are useful.